Hello and welcome to the Plant Party, Native Plants and Insects, hosted by Texas Parks and Wildlife Department, Texas A&M AgriLife Extension Service, and the USDA Natural Resources Conservation Service. Plant Party is a quarterly webinar series intended for advanced plant training and fun education. I'm Tim Sigmund, the Private Lands Program Leader with Texas Parks and Wildlife Department. My co-hosts today are Megan Clayton, Professor and Extension Range Specialist with Texas A&M AgriLife Extension Service, and Charles Kniper, State Resource Conservationist with the USDA Natural Resources Conservation Service. We have 22 wonderful door prizes today we will be giving away as a part of the event. Our door prize donors today are committed to supporting you in your quest to learn more about plants. I'd like to thank Bamert Seed, Indview Range and Pasture, Native American Seed, Texas Quail Forever, Renewable Resources Extension Act, Texas A&M AgriLife Extension Service, Texas Parks and Wildlife Foundation, and the USDA Natural Resources Conservation Service for donating these neat prizes.
All right. Our first presenter today is Stephen Biles. Stephen is an integrated pest management extension agent serving Calhoun, Victoria, and Refugio counties since 2004. He's been an IPM agent since 1998, and prior to that, a field technician with Texas Agricultural Experiment Station. He's a three-time graduate of Texas A&M University and serves an important region for cotton, corn, soybeans, and grain sorghum. The gross annual revenue from these crops in his three counties exceeds $47 million. So he has an important job figuring out how to make them as productive and as profitable as possible with lower environmental impact and lower risk to human health. Lately, Stephen has been a lead researcher investigating a new problematic pasture mealybug, and we're so pleased he could join us today to share everything he knows. Thanks for kicking off our plant party, Stephen.
All right. Thank you very much. So back in October, July of last year was when we first got contacted about this, and I've got quite a bit of information. I'm going to try to get through it as quickly as possible. But we have some other information. If there's something that I don't cover on here, I would encourage you to search Midcoast IPM. That is, I've got two blogs right now. One of them is the old one, and then there's the new one. But you can kind of switch between the two of those and find most of the information I've got today. Pastor Mealybug, July 18th was the first time I really kind of heard about this year. Dr. Jose Santiago Gonzalez in Corpus Christi sent me a text message with these two pictures. This was from the Rio Grande Valley, and it kind of looks bad, but it doesn't look terrible. 2 days later, I got a phone call, or the Victoria Extension Office got a phone call from a hay producer up northwest of Victoria in the Schrader area, and we went and visited there. This was about an 8 acre Bermuda grass pasture that was completely brown, and it really is quite disturbing when we saw that. Just to kind of give you a comparison's sake, the bottom picture is what I expect to see in August, not that top picture that looks like that. The bottom picture was a picture of a research trial I did in 2021. Hopefully, we're going to get it green again. A little background on this: Pasture mealybug comes from Australia. It's been there at least 100 years. You can see the picture on the right is from one of their publications. It appeared in the Caribbean between 2019 and 2020. That was in Puerto Rico and Barbados, where it was a problem. And it causes a problem that the Australians call pasture dieback. Now, I'm going to refer a lot to Australia. They've been studying this for the past 10 or 15 years, and a lot of what we know, in fact, probably 99% of what we know right now is from Australia and their research. Here in Texas, you can see down in the Rio Grande Valley, in 2022, my counterpart, Danielle Sekula, had a comment in one of her newsletters about mealybugs in some Bermuda grass. She noticed that they were also beside sugarcane fields, and there may be an association with that as far as how it came in, but we really don't know. July this year was when I started hearing issues in the Victoria area. Um, and since that time, you can see the map there. Those are counties where we know that it is. We are quite certain it's in a lot of other locations. September 10th of 25 was when USDA positive identified this as Heliococcus somervillei, which is the pasture mealybug from Australia. And that was the first time that it had been identified in North America. Just to kind of tell you how far it's gone, December they found it in some sugarcane in Louisiana, and it was positively identified in early January. So that's kind of the scope of what it looks like. As far as the identification of it, it's a small mealy bug. It's white. The adult females are 2 to 5 millimeters long. They're oval. They have this posterior structure down at the back called anal tubes, and they have these filamentous hairs all around them. The males is what you see up in the top left corner. You're really not going to see those in the field. They're very tiny, and it really, if you see one, you're just going to think it's a gnat. Nymphs can get very small, 0.2 millimeters in length, and that's really kind of the stages I'm looking for right now. It's kind of important to note that all stages of this mealybug are mobile, and that's one of the distinguishing characteristics between that and the Rhodes grass mealybug, which is one that we've had for quite a while, which is really more of an issue in turf grass, like Gulf greens. You're primarily going to find them on the grass blades, but you can also find them in the stems and in the roots. And the females they're often going to leave the plant to lay eggs, either down in the crown. I found them on dry cow patties. You can see this picture on the right. There's about 4 or 5 of them on the underside of this cow pie right there. Life cycle, again, this is information from Australia. It takes about 30 days to go from egg to egg. And they see roughly 3 generations per year, and it will peak in late summer and in early fall. They go through 4 instars after the egg, and a female is going to produce quite a few eggs, 100 or more. The adults live 30 to 45 days. That's going to be something that we're going to keep in mind when we're talking about moving them in hay. And one of the big challenges that we're going to deal with management is that these overwinter in the soil. Video on the right is a hay cutter from one of the guys down around Palacios. And you can see how many there are all over this hay cutter. And moving these from place to place on machinery is probably one of the primary ways where it's moved around South and East Texas. You can also move it on your pants. As you see right there, that's from Danielle. All of the white on her pants are mealy bugs. One of the other ways that it's moved easily around is on mowing machines on roadsides. That's pretty common. Video on the right. I don't know if the video is working, but one of the comments I got from a lot of growers was that my hay cutter is smoking. And this was a guy in the Hallettsville area that he originally thought that he had burned a bearing and it was smoking like that. But all that smoke is from mealybugs. The damage, the adults really don't feed a whole lot. Once the female gets fertilized, it's going to move down to the base of the plant or off the plant, and it doesn't really feed a whole lot. But it's the nymphs that really cause the problem. And you can have one on a leaf, and it can turn that leaf yellow and red, kind of like what you see there in the picture. A lot of the comments that we got from growers was that this one particular pasture it looked weak all year, and we had good hay growing conditions last year, and we shouldn't have had weak or drought stressed looking hay. There's some secondary pathogens that can be a problem, and that's that little picture in the middle that you can see, and that's just kind of secondary to it. And the result is what we're calling an pasture dieback. Just to see some pictures of it, when you get the larger ones, they're really pretty easy to find on the underside of the leaves or on the stems. But in there you see the reddening. That reddening is really kind of one of the keys. When I went into a pasture, I started looking for red areas and looking at those areas first. Just some more pictures of what they look like. Picture on the right is a roadside on Highway 77 between Victoria and Hallettsville. And that kind of demonstrates movement on haying equipment or roadside mowing equipment. Different hosts that I found it on. I specifically found it on Bermuda grass, Bahia grass, and the blue stems. The problem with that is our primary hay species and our primary grass species in our pastures. Johnson grass, we found it on that, and in some cases it was really doing a number on Johnson grass. I was surprised to find it in a hay grazer field. And just kind of a lesson for moving it on your clothes. I moved it into the St. Augustine in my front yard, and really kind of cause some problems there. We know there's a lot of other species. I talked with Dr. Tony Falk with the Cesar Claiborne Wildlife Research Institute. That's the Texas Native Seed guys, and they had a nursery that had like 17 or 18 different native grass species, and most of them, if not all of them, were affected. One of the comments that he made to me was that the grasses that produced more biomass were more affected than the ones that were like more stemmy, and that's kind of a problem. This is an excerpt out of a Australian production as far as how do we get it and what causes the problem. If you have those good growing conditions that you want to make good grass production and you have the mealybug, then you're set up to have pasture dieback. A lot of times, I know some of the early research that I looked at in Australia, they talked about they had mealybugs in a bunch of these fields. Then they got a few rain events, and then it just kind of blew up, and it started causing problems. A lot of the questions I've been is, where are they going to go? We know where they're at now. Where are they going to go? And we don't know the full extent to where they are right now, even. But Australia has, in this particular publication, they mentioned that they find it in areas with more than 600 millimeters of rain. That's 23.5 inches. And if you look at the state of Texas, it's basically the east half of the state. One concerning thing that I did hear or did read last week was there was another publication in Australia that mentions 500 millimeters of inches and more. And that's basically going to move this over into the lighter orange area there. So it could, it could be a significant problem further out west than this line we see here. But we don't know yet. And that's just kind of going to be one of those things that we're going to find out over time. From a management standpoint, I'm really right now suggesting people rely on what we know from Australia, and their discussion is more managed to recover than it is to control the mealybug. Adjust your stocking rates. When you get bare patches, or if you have bare patches, you're going to have to deal with weeds in those areas. A lot of their conversation is shifting some pastures to legumes and tolerant grasses. We don't know what the tolerant grasses are in the United States or in Texas. Fertilizer is going to be important, and things basically to make that, to help that grass to grow better. I know in some situations running a renovator or something like that might actually do some good as far as just mechanically controlling the mealybug. And I've had a lot of people ask about burning and how effective is burning, and burning from the Australian standpoint, they've said that that's been very inconsistent. And the problem is the times that we want to burn, that insect is down in the soil. And so it doesn't have to go very far in the soil to be insulated from that. Some of my thoughts that I've come up with over the past few months, the red and yellow leaves, they're not going to heal. And so once you get that reddening in that leaf, at least that part of the leaf is going to desiccate and go brown. Currently labeled insecticides are not effective. And we did do some trials looking at some of the products, and I'll show you one data slide here in a second. My particular thought is that insecticide control is going to be probably 2, if not 3 applications. And the reason for that is we're going to want to control them. If we get an insecticide we can use, we're going to want to control them in the spring before they become a problem. And that's going to be controlling them before they get reproductive in the spring. And so emergence is going to occur over a period of time, and it's going to require 2 applications to cover all of that time. This is data from the first trial that I put out in Victoria. I put out 5 trials, and the concerning thing is only 2 of them showed any kind of a promise. Part of this, you can see we have the dark green Mustang Max, Carbaryl, which is another product we might use for fall armyworms. Lanate is a fall armyworm type product. I've not used that before, but the challenge with that is even by 14 days, we didn't see a reduction in population. We did see some good effect from Savanto Prime at 8 ounces and at 14 ounces. The challenge with that is 8 ounces is going to be $19 or $20 of chemical. The other challenge is Savanto Prime is not labeled for pasture use in Texas. And so what we're looking at right now is the insecticides that we think may hold promise are going to need label changes before we can use them in Texas. In October, we put out a publication, a fact sheet to try to give some good information. The easiest way to find that is to search Pastor Mealybug and AgriLife together, and it'll come up with this publication, or you can use that QR code on the right. I think it's still active. That's the quickest snapshot I can do, and I think I got that in my allotted time.
Really appreciate that great presentation for that new pest showing up there in South Texas and likely showing up in other places. But with that, we're going to roll right into our second presentation. And next up will be Sam Kieschnick. Sam is an urban wildlife biologist with Texas Parks and Wildlife, serving the east side of the Metroplex. He previously worked as a nature educator with the city of Mansfield at Oliver Nature Park, as a naturalist at the Fort Worth Nature Center and Refuge, as a science interpreter with the Fort Worth Museum of Science and History, as a botanist with Britt, and as an instructor at Weatherford College. He has a master's degree from Tarleton State University, where he studied the genetics of pocket gophers, and currently is an urban wildlife biologist where he focuses on the three A's: awareness, appreciation, and action. Today, Sam is back with us on Plant Party to discuss nocturnal pollinators. I'll turn it over to you, Sam.
Awesome, Tim. Thank you so much for that introduction. We're going to talk about some of the nocturnal pollinators. And these are some of the most exciting things. I think when we go to sleep, nature wakes up. So it is a beautiful, beautiful thing. So first of all, does it even happen? Does nocturnal pollination even occur? Well, yes, it does. Probably not to the extent as it does in the morning or in the daytime. During the daytime, the plants, the flowers will be utilizing the rays of sunshine to make the flowers look pretty, to get more pollinators to come in. So it may not happen as much as it does during the day, but it does still happen at night. There are some really, really fun exceptions to this. There are some vertebrates that do pollination, like our bats that pollinate some of our cacti species in sort of the western part of Texas. And there are some moths and other insects that do it at night. Very crucial here, and part of the plant party is to reinforce this concept that the flower shape indicates what the flower visitor will be. So the flower shape of many of our nighttime pollinated plants typically has fused petals, and they're more tubular. So more tubular flowers, and then also kind of fused petals. Some examples of this that you might be familiar with, the Datura. This is a genus that includes all the moon flowers. Maybe you have some of these in your landscape. Some of the kind of weird ones, this is a group, a genus called the Sclesianthes. These are the trumpets or the angel trumpets, Texas trumpets as well. These are kind of decumbent plants that have these long blooms with a big, long tubular flower. Also, some of our evening primroses in the genus Oenothera, like this Oenothera triloba there on the right side of the picture. Big flowers, tubular, have a tubular base with a little nectar treat there at the base. What sort of things would pollinate these flowers? Well, yeah, these hawk moths. Maybe you've seen some of these sphinx moths and hawk moths flying around. Some of them can be diurnal or active in the day, or crepuscular, sort of evenings and dawn and dusk kind of stuff. But some of these are active at night too. Not a local story, but this is a really fun one to think of. I don't know if you're familiar with this story. This is a orchid from Madagascar, and the species here is Anagrasum sesquipedale. And a guy, a pretty good naturalist named Charles Darwin, looked at this specimen, looked at this plant, and he saw this long flower tube. Didn't see any pollinators himself to it, but he predicted that there was an insect that would pollinate this orchid. Well, about 40 years later, They found specimens of this hawk moth there in Madagascar, of this sphinx moth that had an 11-inch proboscis, an 11-inch tongue. Pretty impressive. And they think it's a symbiotic relationship that this one moth has with this one flower. Crazy interesting. And now I know some of you are in your jammies, which is fantastic. So get comfortable because we're going to talk about the story of this moth, this one in particular moth. This is a geometrid. It's an inchworm, the geo earth metrid measuring. So these are the inch worms. This is an inchworm and it has the genus of Synchlora. Has a couple different names. The emerald moth is a name for it. So Synchlora, maybe you've seen some of these before at your porch or out wherever you may have been. They live pretty much throughout Texas, kind of focusing more on the eastern side of Texas. Most of our urban areas have this specific moth. It's pretty cool. It's pretty interesting. It's pretty. But the caterpillar is particularly interesting. Do you see it? Do you see the caterpillar here? If you look close and if you look at some of these flowers, what this caterpillar does is ridiculous. It goes to a flower, it takes some of the petals and flower parts off, and it glues them to its back. It glues the flower parts onto its back and it continues to eat. So it blends in directly with the flower that it is using. Here's some more pictures of it. Do you see it? Crazy interesting that it does this. Now, there may be some pollination, some indirect pollination that is occurring as it walks over or crawls over the flowers as it's nibbling on some. But man, what a cool bedtime story of this moth. And you may be asking, okay, cool, nice story, but are they even effective pollinators? Well, I would say most of the pollination that most moths do is probably more incidental. So they're not super fuzzy. They're not grabbing a ton of pollen like our bees and wasps and some beetles, those super hairy insects will do. But I think some of it happens just incidentally as they are chilling out on a flower or as they're grabbing some of the nectar from the flower. Yeah, there is some pollen that gets onto their legs, onto their proboscis, onto their foreheads as they visit other plants. So if they're not the best pollinators, should we still value moths? If you said yes, you're absolutely right. We should definitely still value our moths, even though they may not be the supremely good pollinators as beetles and as flies and bees. Why should we value them? They are ecologically crucial. They are so important. This time of year, as birds, even our seed-eating birds like this chickadee, they're shifting their diet. And even the seed-eating birds like chickadees and cardinals, they are going to be utilizing caterpillars to feed their offspring and even to eat themselves. Many of our caterpillars, and I would argue the majority of our caterpillars, are moth caterpillars or grow up to be moths. So birds are grabbing these these larval moths, caterpillars, to feed their babies. So they are absolutely still crucial in the ecosystem. So you may ask, okay, they're not great pollinators, but the birds sure like them. So how should I appreciate some of these moths? I think they're worthy of appreciation, but how do you do it? Step one, diversify your landscape. And I'm not giving you, what do they call it? Financial advice. I'm not giving you financial advice, diversify. I'm giving you ecological advice. So by diversifying your landscape, not only are you making your yard or your landscape more interesting, more intrinsically enjoyable to look at variation, but you're also getting more biodiversity by putting in biodiversity. And some of the biodiversity that I suggest you put in may not be the showiest flowers. The one in the middle here, this is, oh gosh, pellitory, I think is the common name. Parietaria pennsylvania, sometimes called cucumber weed. They taste more cucumber than cucumbers. Really, really cool plant. This is probably not one that you'd submit on a flower competition if you're trying to impress your neighbors. But this little plant, this little quote unquote weed, is a host plant for some very cool moths, as we see on the far right side of the screen. So even some of our interesting moths may come from these quote unquote weeds. So diversifying your landscape is crucially important. Just a little plug here by my good friends, Jim and Lynn Weber. This is a book. I've got a few copies myself. This is The Native Host Plants for Texas Moths. So if you really want to diversify your landscape for the moths and for the birds, diversify it with the plants. So that's step one. Step two, document some of these moths. Document the stuff that you see. This is fun. This gives value to life by learning your neighbors, learning the names of your neighbors. An activity that you can do sometimes, either with a group or by yourself, is blacklighting or mothing, sometimes called. Quite easy. You just get UV lights, ultraviolet lights. These can be party lights. Go to a Halloween store, go to a party store, go online. You can find these relatively inexpensive. This UV light, this black light, has a spectrum that's particularly attractive to nocturnal insects. You put up a white sheet, you put up these black lights, maybe a beverage of choice, and you can watch diversity come to you. And it's one of the most enjoyable activities to do. I don't think I've ever given a presentation in the last 10 years that I haven't mentioned this tool. One way that you can document the moths that you see in your own landscape, in your own yard, in your land, if you have some land, or at your favorite park, is to use iNaturalist. iNaturalist is a citizen science tool. It is where you can document the organisms that you see all around you. Kind of neat with moths, especially if you do it with a white sheet behind it. The visual suggestion, the AI algorithm, the suggested species are pretty good with moths. It's not perfect, but it's pretty good. As you document some of these species, you'll get a suggestion on what the name is. Also, if you use iNaturalist and you want to get verification of some of the plants or bugs or specifically moths, it may take a little bit of time. So patience is necessary if you're looking for verification on those. Another thing too that I'll just offer, if you're used to iNaturalist, if you've played with it before, I will offer my services. So if you want to tag me at the ampersand and then Sam Biology, tag that anywhere, either on the comments or on the notes of your observation. I'll take a look at it. It always makes me so happy to look at these things. And I think it's a beautiful thing that we share our landscape. It's a wonderful thing that we share our landscape with so many different things. I mentioned the story of that Synchlora, that southern emerald, and the crazy interesting caterpillar. That's a simple story. That's one story out of thousands and thousands of stories. In Texas, give or take 5,000 species of moths. So far on iNaturalist, we've documented, give or take, 3,500 different species. So by learning the names of some of these, you can learn the different species that exist out there. And they're just crazy interesting. Despite maybe not being the best pollinators, the pollination maybe being more incidental than on purpose, I think that they are well worthy of appreciation and learning the names of some of them. With that, look at that. Right on time, Tim. Just like we practiced late last night. Thanks so much.
Thank you, Sam. That was great.
All right, excellent. Thanks, Megan. Next up, we have Alan Smith to speak on native pine bark beetles. Alan is an entomologist and forest health program leader with the Texas A&M Forest Service. For 24 years, he has served the citizens of Texas by monitoring and managing for a variety of forest pests and diseases. Alan, it's all yours.
Good morning, everybody. Like you said, my name is Alan Smith. I'm the entomologist and forest health program leader with the Texas A&M Forest Service. For the last 24 years now, I've dealt with native and invasive insects and diseases of our tree species all across the state of Texas. I'm based over here in East Texas, and today I thought we'd spend a few minutes talking about some of our native pine bark beetles and maybe clear up some answers and misconceptions that we have about these particular insects. So first of all of these insects that we're going to talk about today are native to the southeastern United States, including over in western Texas. Most of them, with the exception of the southern pine beetle, are what we call secondary pests, which means they're attracted to and attack and kill stressed trees. The southern pine beetle is an exception. It can go into a healthy tree and kill it. All of these insects feed on the phloem, which is the sleeve of life of that tree where the vascular tissue is. And by feeding and consuming this phloem, the larval galleries, and we'll see images of that in a few minutes, they girdle that phloem and it cuts off the nutrition and water to the tree from the roots and leaves and up and down. And the trees basically starve to death. And this process can happen very, very quickly in some cases in 3 or 4 days. So we have 7 species of these bark beetles in the state of Texas, and they're all very small. Here they are in relationship to the size of a dime. We have 2 genera that we talk about. We have the Ips beetles and we have the Dendroctonus beetles. And they're important because we manage them differently. What we're going to talk about first, and very briefly, and normally this is multi-hours talking about the southern pine beetle, Dendroctonus frontalis, because this historically has been our most destructive insect in the state of Texas and across the southeastern United States. The image that you're seeing here on the right is a single spot that grew over about 9-month period from a 1 acre infestation to almost 8,000 acres. And of the 7 species of bark beetles that we're going to talk about today, only the southern pine beetle has the potential to do something like this. And so that's why it's been on our radar historically and why we spend so much attention to it. These attack weakened, damaged, dying trees, but like I said, they will also go into nice, living, healthy trees. The populations tend to increase and decrease just depending on environmental conditions. And lately, we've seen intensive droughts with a lot of other tree stressors, but the intensive drought and high heat have really hammered these beetles, and they have not been a problem in the state of Texas for several years now, which is good news. The map that you're seeing here shows the hazard associated with southern pine beetle, where if it was to appear, we would expect to see large amounts of pine mortality. And this map is basically built off of host density, host conditions that are attractive to the southern pine beetle. We trap for this insect every year. My traps are out in the woods right now. Our data from 2025 shows that we caught 0 southern pine beetle, which is great news, which means that our probability of running into that insect in the woods this year is very low. That's good news. The disturbing news is that the clarid beetles, which is their main predator, we only caught 3,600 of those. And last year, you can see in the graph in 2024, our trapping data, we caught over 11,000 clarids. So that decrease in clarid population has us a little concerned that their main predator may be not in large enough numbers to control the southern pine beetle. But the good news is, with no southern pine beetle caught in our traps or any of our monitoring stations, we feel fairly confident we're out of the woods, so to speak, with southern pine beetle for this year. All right, so let's talk about Ips beetles, because that's the main vector of mortality that we're seeing across our pine landscapes in Texas right now. We have 4 species of Ips beetles in Texas. Ips pinei is a western species. We find that out in the Davis Mountains and the Guadalupe Mountains, but Calligraphus, grandiculus, and avulsus are species that we run into in East Texas all the time. These are very common in the southern United States, southeastern U.S. They will go into weakened, stressed, and damaged trees. Their antennae are specifically tuned to pick up those weak tree pheromones. And unlike the southern pine beetle, their biology is the male starts this process where he finds a wounded, stressed tree, digs himself a nuptial chamber, releases pheromones that bring females to him. They mate in this nuptial chamber, and then each of the females then crawls down, creates a gallery, and lays her eggs along the sides of her gallery. And it's these galleries and the developing larval galleries from these eggs that cause the girdling and mortality of the tree. And it's pretty interesting. We can read these gallery patterns just like a fingerprint. So we can pull bark off of a dead, dying tree and read it like a fingerprint, and it will tell us exactly what species of Ips beetle caused that mortality. These insects have many overlapping generations per year. In fact, Ips avulsus, which is our smallest one, they can complete their development and life cycle in about 2 weeks, and thus have about 10 generations, and sometimes in more, depending on environmental conditions per year. These insects have a very, very high threshold for heat. They love it at around 102, 103 degrees, and their biology just kicks off, and they produce more eggs, and the larvae survive and grow bigger. They disperse further, and they become a bigger problem as the heat increases. Oftentimes, these attacks are very small when they initiate. It's a one little small part of a dead tree. That branch may end up dying off, and then as the beetles populate that tree, they go into the bole or the trunk of that tree and then start to girdle that and then kill the tree from there. So what do you look for? Well, you look for branches starting to flag or change color. We always look up at the crown of the tree, the very top, and what we want to see is a dark green healthy tree. And when we see it starting to change color, that tells us that tree is having problems. And when we see that color change go from dark green to light green to yellow, orange, red, that's the mortality progression. And that happens from the top downward of that tree as that vascular system is compromised, and the beetle and the tree cannot move its water up to where it needs to be in the top of the crown. Usually by the time you see orange, yellow, orange, and red, that tree is dead, and there's no saving it no matter what you do. And it just becomes a matter of removing it for safety concerns. On the outside of the tree, you might see in the center image what we call a pitch tube. That's the tree's attempt to keep the beetle out. That will only occur if the tree has enough moisture to produce sap. Lately, with high drought conditions, I've seen a lot of Ips beetles killing trees with no external sign, simply because the trees don't have enough sap to fight back. Peel the bark back, you can see the galleries. Like a felled tree here, you can see large piles of boring dust as these beetles kick the dust out of their hole as they excavate their galleries. Like I said before, these occur on stressed and injured trees, and that stress can be a natural process. Might be a wind damage or a hurricane, lightning strikes. We see prescribed burns and natural wildfires stressing these trees out and causing a weakness and attracting beetles. Anything mechanical, people building sidewalks and swimming pools next to roots, to skidders and logging equipment, to motorbikes crashing into the stem of the tree and scraping the bark off, releasing those wound pheromones. But our big ones are drought and heat, and those are the main stressors that we're seeing in our pine trees right now. Typically, what we see when we find, unlike that giant spot that I showed you with southern pine beetle, the Ips mortality is very spots, very individual tree oriented. There'll be one tree over on one side of the stand, one tree in the middle, one tree over there, and it only picks the weakest. What we've seen over the last several years is more of a concentrated effort as all the trees in that particular stand get weak at the same kind of rate, and the beetles don't have to fly across the stand and pick their victims. They can pull them right here and take a whole cluster of trees. Lastly, we have the turpentine beetles. These are species that produce large globular pitch tubes at the base of the tree, probably the last bottom 10 to 14 feet of the tree. They are our largest species, but the least aggressive. And again, they have the same kind of mortality attack pattern, but they make these large purplish globules of sap near the base of the tree, and they're pretty easy to identify. So what do? Well, grow healthy trees, keep them happy, reduce their stress, increase their vigor. And if all else fails, well, then we've got to go in and we've got to do some treatment. And what we can do is we can do a preventative bark spray, which isn't a super great option because it's really impossible to get the entire tree coated with an insecticide. The insects that are causing the damage are inside the tree under the bark. And so the insecticide and the insects really don't come into extended contact. And it produces a lot of overspray that your neighbors and your pets really won't appreciate. We can do a soil injection of different chemicals where we allow the tree to uptake that pesticide and spread it throughout its own system internally. This has some drawbacks and some advantages. It's cheap, but it takes a long time for it to spread. And if the tree has been compromised with bark beetles already, the vascular system isn't working as well as it should, and it won't translocate the chemical as efficiently as we would hope. The other option we can do, of course, is inject an insecticide directly into that tree. And it has a lot of benefits there with worker safety, lower dosages, things like that. But again, it relies on that tree being healthy enough to translocate the chemical. It is also quite expensive. The chemical that we use here, triage formulation with imamectin benzoate, is extremely expensive, but it does last a long time, and it is very effective on bark beetles in pine trees. Lastly, I want to mention this insect. I get calls all the time that I can hear beetles chewing on my trees, and it's a pine beetle. Well, it's a pine sawyer, which is a large cerambycid longhorn beetle. And the good news is you're not hearing pine beetles chewing on your tree. The bad news is this is the cleanup crew, and they're coming in to get rid of the dead, dying pine tree. So if you can hear beetles chewing in the tree, this is what you're looking at. Large holes on the outside of the tree, big egg niches, and you can contrast it there with the southern pine beetle pitch tube, or with the Ips beetle pitch tube in this case. So lastly, make sure, we don't need to fear every single beetle that you see out there in the woods. They're not all scary, but a little knowledge goes a long way in saving you money and heartache and allowing you to manage your pine trees for these bark beetle problems. So I've got my contact information right here. Thank you very much.
Thanks, Alan. That was a great presentation. So next up, we'll be moving on to monarchs. Katie Harris serves as the Xerces Society Insect Pollinator Conservation Specialist and Partner Biologist with the NRCS Texas State Office in Temple. Katie received her MS degree in Environmental Science and Policy from the University of Wisconsin Green Bay, where she conducted a biodiversity survey of spiders. Katie began studying and creating effective monarch butterfly habitat in 2018 and graduated from the University of Missouri in 2023 with a PhD in plant, insect, and microbial sciences. Prior to joining the Xerces Society, she worked as a postdoctoral researcher with the University of Texas at Austin, examining the effects of parasitism on dung beetle ecosystem services in central Texas. Outside of work, you can find Katie spending time with her parrots and riding horses. Welcome to the plant party, Katie. I'll turn it over to you to tell us all about oleander aphids on milkweed, and are they monarch enemies or allies?
Wonderful. Thank you so much for the introduction, Tim. I'm so happy to be here today for Plant Party. And there we go, Plant Party. I thought it was only fitting for all of the insects to wear party hats. So today I would like to talk a little bit about some of my dissertation research that I conducted before joining Xerces and sort of how these oleander aphids interact with monarchs in the wild. I tend to get a lot of questions on this, so I'm excited to share some of the findings. So essentially, monarchs are in decline in North America, and we want to preserve this unique behavior that they have of this migration. So it's really important to understand their resource needs. So especially here in Texas, this would mean providing nectar or wildflowers to them in the springtime so that they can fuel their flight, and also again in the fall when they migrate south again. For overwintering is when they need to store up all those lipids to be able to handle overwintering. In the spring, another resource that they need is milkweed. Milkweed is so important for their life cycle. The adult monarchs lay eggs on milkweed plants and they grow into caterpillars and they'll pupate, and that's how they complete their life cycle. But there's other insects that are also present on milkweed as well, such as oleander aphids. And if any of you have gardens and whatnot, and I bet you have seen these before, they are little phloem feeding insects, which means they essentially have this little stylet, it's called. It's like a little straw that they stick right into the plant stem, leaves, or flower heads, or seed pods, and they'll basically suck out sugar water. And they can reach quite high densities. They're in the summertime, especially, they are female and they reproduce asexually. And when they're essentially born, they already have embryos for the next generation and they give live birth so they can replicate real fast. So most of the time when we think of aphids on milkweed, we think, oh my gosh, pest insects, their excretions or their poop essentially is a sugary, sticky substance that if it builds up on plant leaves, it can cause sooty mold. And they're using the same resources as monarchs. So are they competitors? Are these aphids actually bad for monarchs? So there's a few objectives I'm going to try to get through very quickly today. But essentially, I want to look at how oleander aphids affect the monarch egg-laying behavior or oviposition, how egg predation also plays into this, and also how the aphids can affect nectar quality. So this all stems from some research I conducted at the University of Missouri under Dr. Deborah Finke. It was based off of this large field study where we were looking at effective habitat for monarch butterflies. So where should we put the milkweed along the landscape, and what kind of resources should surround the milkweed for optimal habitat? But what we, I have more information on that as well. That paper has been published, and if you're interested in those findings, I'm happy to share those as well. But I want to get into the oleander aphid aspect, because while we were conducting this research, we found that there was more monarch eggs on plants where more aphids were present. So we're like, what is going on here? I thought that these were like pest insects for monarchs. So in order to kind of figure this out and tease it apart, I did some greenhouse studies. So essentially controlled experiments where like there's no predation or other variables that are so prevalent in field studies. And this is just a video of an example of when I was testing methodology for these studies. I essentially take a mated female and stick her in a very large cage with a plant that has no aphids on it, a plant that had a low number of aphids on it, and a plant that had a high number of aphids on it, and let her choose where to lay her eggs. So plotted, this looks, it's kind of like quadratic in nature, right? So at lower aphid densities or numbers, we saw this positive association once again, where there were more monarch eggs on plants with aphids. However, it seems like there reaches a certain high density of aphids where the monarchs choose not to lay their eggs on these plants anymore. So why could this be? Why is there a positive interaction between monarch eggs and aphids? Well, there are some studies that show differences in plant chemistry. So when insects feed on plants, oftentimes the plant will defend itself with chemicals. And aphids, the ones with the straw mouths, have a different feeding strategy than caterpillars, which are more of like the chewing type. And this can induce a different mechanism that benefits the caterpillar chewing. So essentially, the plant's defending against the aphids, and it's allowing the caterpillars to feed a little bit more readily. So that's one theory, is that perhaps that there's some chemical trade-off benefiting these caterpillars. And then there's other factors as well that could be occurring. Perhaps the aphid honeydew at lower levels. It's basically sugar water. It tends to be attractive to various types of pollinators. Maybe the yellow color of the aphids. It's all very unknown at this point. But in terms of the chemistry trade-off, this is likely to also vary by species. So we do need more research in this area to study different species of milkweed, but in common milkweed, and then swamp milkweed was another species that I studied as well, it seems like there's that positive association and potential trade-off of benefiting larval caterpillars. But for tuberosa or butterfly milkweed, researchers didn't find that chemical benefit for the monarchs. And there was a recent study done on tropical milkweed with higher densities of aphids, and they also found that the aphids were not that good for monarchs. So we saw that positive association at lower numbers of aphids, but why would monarchs not choose to lay their eggs on plants with higher densities of aphids? Well, a couple theories here. Plant quality, perhaps. They're using the same resource as monarchs, and the plants, sometimes when they get really high densities of aphids on them, they start to like wilt a bit and look look a little bit more sad. Maybe spatial limitations. There's so many aphids that are covering the plant. Maybe the monarch isn't laying their eggs on the plant because it can't reach the plant. And then one more sort of silly idea is that maybe some of these eggs are walking away on aphid backs, and that's why they're not being counted in these experiments. I've noticed this a few times actually in the field, and I've seen it posted elsewhere as well, where monarchs, there's so many aphids on the plant that they'll lay the egg right on the aphid back. So the second objective I wanted to bring you through was the potential for alternative prey. So are these oleander aphids more likely to get eaten by, say, a lady beetle predator, or are they going to get, or are the eggs going to be predated upon? So I ran some Petri dish trials. Essentially, I put eggs, monarch eggs, in Petri dishes with and without aphids present. And as you can see on the left here, when aphids were absent, pretty much every time all 3 of the monarch eggs were consumed. But when aphids were present along with the monarch eggs, then the occurrences of monarch eggs being eaten were a lot less. So these aphids being on the plant could potentially help protect the monarch eggs, and also the larvae from being eaten. All right, and then the last objective I wanted to take you through really quickly was floral resources. So these aphids are feeding on these milkweed plants, and they're just taking all that sugar water right out of them. So nectar is essentially sugar water. So how are the oleander aphids potentially affecting this nectar quality for adult monarch butterflies? And quality is denoted by like sugar concentration and the total volume of nectar produced. So in order to assess this, I essentially grew up these swamp milkweed plants in the greenhouse. You can see on the left there, I have a graduated cylinder in my hand. I was measuring perfectly the amount of water, and they were each gotten the same amount of water. And I had to do this 2 or 3 times a day for several months until the plants could reach a flowering stage. And then I applied a treatment of no aphids, a low number of aphids, and a high number of aphids. So then when it was time to extract the nectar, I had to very carefully brush off every single aphid on the treatments that had aphids from the flowers, and I put them in a little tube with some glass wool, and I essentially spun them really fast to get all the nectar out of the flowers. And I tested the sugar concentration with a refractometer, and I measured the total volume of flower nectar as well based on these treatments. And essentially what I found was that there was no difference in sugar concentration between the treatments. So essentially the aphids weren't affecting the concentration of sugar that the plants could put out in their nectar. But we did find that there was a significantly less flower nectar produced by flowers that had a high density of aphids feeding on them. So there's some implications involved with this. So there was no change in sugar concentration, but the total volume of the nectar was reduced. So this has implications for potentially milkweed visitation. Are the monarchs going to visit these flowers less often? Or are they going to spend less time on these flowers? And then as well as adult fuel. So the monarchs need this nectar as a resource. And if less is being produced, then that could be harmful to them. So in conclusion, are aphids enemies or allies to the monarch? And it truly depends. It seems like at low to moderate densities in some species of milkweed, like common and swamp milkweed, there is a potential positive for monarchs. They're able to consume, the caterpillars are able to consume the plant material more readily. But at higher densities, this is likely a negative. They're taking down the plant quality, taking up space on the plant, and there aren't any good chemical benefits anymore when their densities are so high. In terms of alternative prey, it appears as though they can serve as alternative prey for predators like lady beetles. So when a lady beetle's bopping around on a plant, it might be more likely to eat an aphid than it would a monarch egg. And then in terms of the nectar quality, it seems like the aphids are negatively affecting monarchs at high densities for the flower nectar total volume. But of course, more research is needed. It would be wonderful to have some of the more Texas species studied as well, like Viridis, Onthoroides, and Asperula. So in conclusion, this is usually what I say when people ask about what to do about oleander aphids. And I usually say, well, it's okay to leave them, especially at lower numbers, because there may be in some species of plants, there may be some benefits to the larval caterpillar, but also there are a lot of natural enemies, predator insects that will take down their numbers. Sometimes with aphids, they'll reach this critical density and then they'll just sort of disappear because their densities have reached so high that they either move on or they die out on their own. I would recommend avoiding pesticide use, especially since you might accidentally get a monarch or some other beneficial insects. But if you're really desperate to get rid of the aphids off of your plants, I would recommend to spray them or gently wash them with water. You might have to use a little bit high pressure in order to get the aphids off with just water, but that's a very gentle way to do it. Just make sure there aren't any monarchs present if possible. All right. And then thank you so much. My contact information is here if you want to chat more about this or any of the other similar research I've done. Thanks so much for having me.
Thank you so much, Katie. That's really interesting research. Excellent presentation. All right, so we are ready to get on with our last presentation. Our final speaker is Dr. Tyler Gilreath. Dr. Gilreath is an extension entomologist with Texas A&M AgriLife Extension based out of Canyon, where his work focuses on the management of economically important insect pests and field crops. His research and extension programs emphasize integrated pest management strategies for pests of corn, cotton, wheat, and sorghum, with particular attention to insecticide efficacy, resistance management, and on-farm applied research. Tyler works closely with producers, extension agents, and industry partners to translate research findings into practical, field-ready recommendations for Texas agriculture. I'll turn it over to you, Dr. Gilreath, to speak on desert termites, something I wish I had $1 for every time I'm asked about, recognizing damage and managing risk.
Well, thank you for that introduction. Like she said, my name's Tyler Gilreath. I'm the extension entomologist now based out of Canyon. Used to be out of Amarillo, but we recently moved. As I was doing my graduate work with David Kearns out of College Station, we had a number of calls looking at desert termites and questions about it, what do we do and how do we mitigate the situation? The funny thing about the whole thing was my parents, which live in the Hillsboro area, actually dealt with this personally. And so I had a pretty good field site to look at these things and try to get some answers. And the short of it is we really don't know still. But, we did put a trial out and trying to see kind of where we were at with it and how to answer the questions or figure out what's available at this point. So this is what we were looking at. For those of you who don't know, this is very common in pastures, and I think people overlook it. People don't know what it is. You see the tunneling, across the ground. You'll look at some of those, the grass blades over there on the left, looking at tunnels going up the leaf blades themselves. And then during parts of the day, depending on kind of when you're out and about, you'll see them foraging on the ground. They are a termite species that likes to be hidden. When it gets too hot, they kind of go back into their tunnels, but that's what they're leaving. And, on worst case scenarios, this is actually a personal photos that I got to take on some family ground. You can see that it can be pretty devastating. We've seen quite a few pastures get taken to this pest with no regards of how to fix it. And so that's kind of where we came in and kind of tried to understand more about this pest and figure out what it is. When we look at the occurrence and damage, they're primarily feeding on dry grass. They don't like green grass. They don't want to feed on it. A lot of guys have called, and we've gotten several out of the far West Texas area saying, hey, I've seen this pest. It's all over my fence posts. Are they eating my fence posts? And no, they don't like wood or anything like that. They really prefer the dry grass. And essentially what they do is they're building those dirt tunnels or they're, over the green grass, which is eventually killing it so that they can feed on it. We have found them on a number of different things. They seem to really enjoy cow droppings. You can flip a lot of those over in your pasture and find a number of these individuals underneath it, just kind of foraging about. But really they focus on dry grass, which of course we all have seen that. If you've ever ran into them, you know that. when we talk about populations and density sizes, this is one of the most difficult things. And actually, we got to talk with Dr. Robert Puckett, who is our urban entomologist, because he deals a lot with termites, and we kind of teamed up with him to try to understand if there any similarities. And we didn't get real far with that. I don't think there's a whole lot of similarities really, other than they're just a termite species. But when we look at how dense they could be, anywhere from 4,000 termites in a cubic meter, so somewhere in that ballpark. They can consume quite a bit of plant material in a day, depending on population sizes. They seem to kind of build their populations in a wet year. When it goes from wet into a dry conditions, that's when we really started to see them. Really, and my personal experience with them is when you had those dry spells and the pasture kind of starts dying back, that's when these things kind of took over of them. And they won't outcompete a grass species, and that seems to be kind of, one of our best ways to mitigate this issue. But they can be a problem. Really, when you're following a wet year, that's when we got some of our larger populations to pop out. And so we got a trial out, like I said, on some family land, really with there's nothing in the literature that suggests how to control them. We had a lot of growers and a lot of guys contact us, and malathion or something like that was commonly used as a pasture insecticide anyways, was kind of the question, hey, does this work or does it not work? So how do we do it? Well, we didn't know where to start. I know that my family had been told from older generations that, running a drag harrow like you see in that bottom left picture seems to mitigate them, seems to knock them down, and that could kill them. So what we came up with was basically an insecticide trial where we wanted to look at different insecticides, whether we harrowed or we didn't harrow, and how great of control could we get on it. And interestingly, what we used was an app on our phone. You can see those are the pictures on the right are kind of what we did. So that's just basically a 2 foot by 2 foot square, I believe is what it was. And then you took a picture with the Canopio app. And I think Stephen Biles, I missed his presentation earlier. I'm sure he probably mentioned it. The same one where it's basically % green up, and see how good of a control we can get. Now, it gets a little more complicated and convoluted when you consider that this is, a pasture setup. So green up could be based off of how much precipitation we got, soil types, and there's a number of things. But this is kind of where we started, just to get an idea on how well of control could we get of these things. And so when we looked at harrowing specifically, really by itself, it didn't seem to add just a whole heck of a lot to it. There was no significant impact. I know there's still a bunch of old guys that are still going to run that. They think it does. The harrowing by itself seemed to kind of give you some control just by killing them, just because you're knocking down and they're having to rebuild those tunnels again. But what we saw on the flip side, on what our data suggested, is that actually harrowing and disturbing that pasture seemed to kind of slow down the forage regrowth. And that could be more problematic than beneficial in a lot of situations. So some things to keep in mind as you kind of consider treating these guys and figuring that out, which again can still be difficult. The other thing that makes it very difficult for us is the amount of insecticides, and I know Stephen Biles is running into this when we're starting to control the pasture mealybug, is there's not a lot of options really out there as far as pasture insecticides go. We know we have a lot of herbicides, but we don't have a lot of insecticides that work very well or don't provide a lot of different modes of action by any means. You can see we only have a handful of different, IRAC groups that are available. So really what we did was pick a few of them that we thought would work the best, and let's see how well it works in a pasture setting. So this is what we did. And again, this is some older data. This is from 2022. We're looking at Vanacore, which is a very widely used product across the row crop market. The reason why we liked Vanacore is because it offers some translaminar activity. So it will move within the leaf. If you spray it on the top side, it moves to the bottom side. So effectively, if you can't get good coverage, you're expecting the insecticide to help you out with that one. And then a couple of other ones, looking at a couple of pyrethroids, Intrepid, malathion, because it was one of the most commonly used. and of course lambda-ci as well. And looking at the data, percent control, and this is just based off of tunnel coverage. And so, and again, it's difficult to count an insect. And most of the times when we do an efficacy trial, it's counting the number of insects that are either dead or not present, and would give you some idea of control. But it's a little more difficult when you're talking about an insect that we can't actually see because their colonies are underneath the surface of the ground. So it made it a little difficult. And this is the best we could kind of come up with was, can we look at it on tunnel coverage and how good of control we got? And you can see really not a whole lot of efficacy from the harrowing itself at 15 days. We do see some activity with some of those insecticides that maybe offer some of that control. One warning there is that Vantacor now, it's going to be fairly pricey. There are a few generic versions that might make that more attractive by any means. When we got it to 29 days, again, it's kind of the same setup, not a whole lot of differences there. One important thing that I really like to point out is when you start using products like lambda-ci, which is a pyrethroid, just a very broad insecticide, most of the time we see a backwards effect. But it seemed to be working here best for us. Now, it doesn't give us a lot of residual activity, but it did seem to work here. It's not one that we like to use because of how broad spectrum it is. It's going to knock out a lot of beneficials, but it does seem to work here for the most part. But again, it's only going to last you for a little while. And again, I know it looks like it lasts a while here in our trial, but it's hard to say so without being able to really understand how much control we're getting from these products on that insect that we can't actually count. And it gets difficult. And this is really the best data that we've got. And again, I hate sharing it because we don't know. The product, the sites that we chose, they kind of just vanished for a little while, and then some other pests popped up, and we really haven't had much more data recently. And insecticides seem to work, here or there. I know that the recommendations that we made, so we put out a trial here on one little spot. I told my dad to go ahead and spray the rest of his pastures using some other things, and they got mixed results depending on where we were at. They weren't the same results that we saw here. So again, very variable with the results of the data, but again, it's very difficult to try to control this insect, and really controlling it with insecticides doesn't seem to be the most effective. We just don't know right now. I hope this plays for you. This is a success story. So just a little backstory on this one right quick. This entire pasture right here that you're seeing was completely ate up. This is actually right where that trial was at. You can see the previous pictures that you saw, that was this pasture where it was very bare, a lot of tunneling everywhere. But now this is a very recent, I think this was about a month ago. A month ago I asked my stepdad. So this would be probably last summer that this is what this looked like here. So we do have success. Now, one of the hardest parts, and again, the hardest part that we found with controlling these things is the variability within the insecticides, and then harrowing seemed to provide very little control. The idea with the harrowing that we tried or wanted to do was, if you could open up those tunnels, can we access them to get an insecticide to them? I just don't think that that's what we ended up achieving necessarily. And in fact, I think, as our data suggested, that it caused more damage to the pasture than it helped. But again, with those limited number of insecticides available, it makes it difficult to really control these. The most optimal thing in order to control them is to outcompete them. What we found that works the best was that if you can get those wet years back and get your pasture back to a greened up material, making it look where you can outcompete them, that seemed to be the best. But again, it comes difficult when you're talking about pasture or any sort of rangeland. I mean, obviously, we can't dictate what Mother Nature is going to do. And, if we get rains, we get rains. If we don't, it kind of gets a little more difficult. But where we have seen the greatest success, I would say, is when we went into a complete failure of a pasture due to termites, able to go in and resprig those pastures and then start out competing them, starting to plant. I think we put a couple different Bermuda grass varieties out that established very well. They established very quickly, and that was able to provide a level of control that we were satisfied with, right? And so being able to outcompete them seems to be the best way. If you get into a dire situation, again, we do have some insecticide options to consider. Again, it's going to be based on economics of trying to get over the top of that pasture or rangeland. And then really, you know, I think one of the biggest things we don't fully understand is what's the economic value or economic impact that these insects have. Again, there's not much in the literature that suggests. You can go back and find where people were talking about them, but we don't have really economical impacts of them. So we don't know at what point do we you know, when do we trigger? What's the threshold for these guys? When do we need to spray? That we don't know. And that's kind of difficult again with the limited amount of work we've done with them. It gets a little more difficult. But again, I would say that the most promising thing that we have is really out competing them and trying to get that keep that pasture healthy. I know that my parents are very, very rigorous when they put fertilizer out. We're trying to get it out timely before rain so that grass has the optimal growth potential that we can really manage and try to outcompete them. I don't know if Stephen Biles covered this or not. Again, I missed his previous talk, but every week we do some updates. Y'all might be interested. If they're not just going to be row crops. Obviously, we have a couple of new alerts for row crop guys, but you can scan the QR code and keep up to date. Occasionally, I say occasionally, more often than not. Obviously, you'll get some updates from some Stephen Biles on the pasture mealybug. You'll get some of those. David Kearns will do a lot. He'll put out weekly updates, and if there's something, a big hot topic, if there's a big push of fall armyworms coming through pastures, you know, he'll put it in those weekly updates and kind of discuss some of those options. So it's a good way to stay connected with folks and a good way to hear kind of what we're seeing on the IPM side and the entomology side. There's a couple of different regions you can hit up. But yeah, sign up for that. They're just quick, you know, once a week during the growing season, we put these out, and an easy way to kind of stay up to date with where we're at and what we're doing. And then that's kind of all I got. If you feel like you need me or if you want to get a hold of me, here's my contact info. Feel free to reach out to me. Let me know. I'll be more than happy to try to answer the best I can. Again, we don't have a lot of experience or data with it. And if I can't answer it, I'll get you in touch with David Kearns. I know he has a lot more about it than I do. Probably knows a lot more about it than I do as well. So feel free to reach out and contact me, and I'll get you an answer one way or another. So.
All right. Thank you, Dr. Gilreath, for that great presentation. Want to take again the opportunity to thank our door prize sponsors of Bamert Seed, NG Range and Pasture, Native American Seed, Texas Quail Forever, Renewable Resources Extension Act, Texas A&M AgriLife Extension Service, Texas Parks and Wildlife Foundation, and the USDA Natural Resources Conservation Service. We appreciate the support for Plant Party. We want to take the opportunity again to thank all of our speakers, and of course we want to thank you for attending the 22nd Plant Party webinar. Again, Plant Party is a quarterly advanced plant webinar training series brought to you by USDA Natural Resources Conservation Service, Texas Parks and Wildlife Department, and Texas A&M AgriLife Extension Service. We'll be sending a link to a short survey by email, or you can type in the address you see there on the screen and chat box, or we'll pop up the QR code that you can simply hold your phone camera over and click the link that pops up. Mark your calendars for the next Plant Party on June 24th. We'll be covering grasses for the wind. Be there or be like the stems of mint. Thanks, everybody.